So now we're on to our next topic, which is transcription in prokaryotes. So first thing we have to establish is that in prokaryotes, we have a region, or transcription only happens based on a single strand of DNA. So for example, if you have your DNA strand here and another one here, so if this is your 5' to 3' strand, and this is your 3' to the 5' strand, so remember that nucleic acids are always synthesized in the 5' to the 3' direction. As such, it would make more sense for this strand here at the bottom to be the template strand. So, if this is a template strand, this is also known as the non-coding strand or the antisense strand. Or sometimes people would say it's the negative sense strand. Whereas this strand on top is called the non-template strand. The coding strand. The sense strand, or the positive sense strand. Now, this strand here will have all of those names. It's called the coding strand or the sense strand because eventually the mRNA that you synthesize based on this strand will have an exact same sequence as this strand here, except that the T's are now replaced with U's. As such, this is a sense strand because it's the sequence that makes sense in terms of proteins. So, usually, sequences shown in transcription are the sequences of the sense strand, even if, sa totoo lang talaga, this is the template strand. Ito yung strand that participates in transcription. Okay, I hope that that's clear. So anyway, on our DNA, there will be several regions that are important for transcription, and that region is called the promoter region. So this region is important because it quote-unquote promotes transcription. So kung walang promoter region, walang transcription na magaganap. So the promoter will have several parts. It will have the transcription start site, which is shown here as positive 1. So, pag sinabing positive 1, ibig sabihin siya yung pinakaunang base na makikita ninyo sa inyong transcript or sa inyong mRNA. Upstream of that, so pag sinabing upstream, yan yung mga bases before a reference. So, in this case, kung ito yung reference natin, anything upstream or anything before that is upstream, whereas anything downward would be downstream. Ang term. So, in the upstream region, we have the Pribno box. So, if you will find the Pribno box is rich in A's and T's. And this is conserved across several genes and across several species. So, ito siya, medyo consistent siya na 10 bases upstream. So, pag may nakita kayong minus 10, ibig sabihin 10 bases siya upstream ng reference natin. Na usually, ang reference is the transcription start site. So, we have the Pribno box, and upstream the Pribno box is the minus 35 region. So, itong parts na to, ito sila, they are largely called the core promoter. Ibig sabihin, sila yung pinaka-important parts ng promoter mo. Pwedeng kasi sa promoter mo, kung ito yung promoter region, pwedeng sa before niyan, may mga enhancer regions pa or whatever. So, itong mga regions na to, they enhance transcription. So, ito sila all together. Pag pinagsama, ang tawag dyan ay extended promoter. So, sa extended promoter mo, important siya because it will enhance transcription. Pero, kapag wala ito sila at core promoter lang meron, transcription will still occur. Pero, kapag walang core promoter, walang transcription at all. Hindi mangyayari ang transcription. Okay? So, itong promoter, siya ang nire-recognize ng sigma factor. Siya yung shown dito as green. So, the sigma factor is a subunit of RNA polymerase. So, in prokaryotes, you only have one RNA polymerase, kaya wala siyang number. Kasi nag-iisa lang siya. So, whether ang product mo ay mRNA or rRNA or tRNA, isang RNA polymerase lang ang nag-produce yan lahat. So, si sigma factor, important siya kasi it recognizes the promoter region. So, once sigma factor recognizes that, the RNA polymerase comes in and binds to that region. Once they bind to that region, you form a closed promoter complex. Okay? So, once your closed promoter complex has been formed, it is now ready for transcription to start. So, if you have an energy source that comes in, this part becomes an open complex or an open promoter complex. As in this part of the promoter, which if you will remember is AT rich, starts to unwind. And so, nucleoside triphosphates can be incorporated. So here, the nucleoside triphosphates are incorporated. So this is now your mRNA, your starting mRNA. So this will be your +1, as in the transcription start site. Tapos, you will just continue on adding the nucleoside triphosphates. Ayan. Until, at some point, your sigma factor dissociates. Sigma factor is not necessary for the elongation of your mRNA because its main purpose is for recognition. As in recognition of the promoter region lang talaga. So, after a few bases into the mRNA, the sigma factor detaches and it can find a new promoter to recognize and once again start transcription there. So, it's kind of recycled. So, this elongation continues on. So, sa transcription, we will be involving topoisomerases. Largely because if you unwind your DNA, there will be torsional strain here and here. As such, topoisomerases are important to relieve that torsional strain. Okay, so if we terminate transcription, there are two ways of terminating it. It can either be raw dependent. So, this letter here, this is raw. This is not letter P ha. This is raw. Because this refers to the raw factor, which is a protein that helps with termination. So, in this first termination type, it's intrinsic termination or also known as raw independent termination. So, in this case, you have your DNA and they have regions that are complementary to each other. So, because they are complementary, if you synthesize an mRNA, which is single-stranded, the effect is that that mRNA forms a hairpin loop. Once it forms a hairpin loop, it also has a region that is rich with uracil. So meaning if it's rich with uracil here, it binds to a DNA that is rich in adenine. Meaning, this is sustained by 2 hydrogen bonds only, which are weaker bonds compared to your GC. As such, this mRNA falls off of the DNA template. So far, they don't really know the exact mechanism how this falling off happens. It's just that they assume that because this is the weaker hydrogen pairing compared to GC, that somehow it can just fall off. So if we have a raw independent termination, we also have a raw dependent termination. So with raw dependent, it involves the raw factor, this donut-looking flower here. So the raw factor facilitates the dissociation of the RNA polymerase and the mRNA from your template DNA, because the raw factor is a cousin of helicase, which, if you will remember, functions in unwinding the DNA in replication. So the raw factor basically just spins them. So it unwinds the mRNA and then it helps in its removal. So I have a video of this, which I will show you. So it's a video on transcription in prokaryotes. So this is a prokaryote. These are all its other regions. This is your DNA. So if you can see, this is your Pribno box, which is found in the -10 region and your -35 region. So this is your promoter, which is recognized by the RNA polymerase and the sigma factor. So you have now a closed promoter complex, and then it becomes an open promoter complex. So if you can see, the DNA has now unwound. And then now we've located the transcription start site, shown as +1. And now the nucleotides here are incorporated. So these are RNA nucleotides. And then it's elongated. So if you will remember, prokaryotes do not have a nuclear membrane. Because of that, the mRNA can be translated right away. So this process is called co-translation, meaning sabay ang ating transcription sa translation in prokaryotes. So now, in raw independent termination, if you will see, this hairpin loop has formed. The presence of this hairpin loop stalls or stops the RNA polymerase from moving forward. And then this part here, the U-rich region and the A-rich region, because these are weaker bonds, it will help the mRNA from dissociating. It will help the DNA, the RNA rather, dissociate from the DNA template because of its weaker bonds. Now, in raw dependent termination, the raw factor, which is this thing here, comes in, and then there. A hairpin loop is formed once again. There you go. Hairpin loop. And this hairpin loop stalls or stops the RNA polymerase from continuing on. And then because of the raw factor, the mRNA and the polymerase dissociates from the template. Okay, so let's continue on the slide. So here shown is an operon. So an operon is basically this entire stretch. So you have a promoter and 3 genes that are under its control, and this promoter is under the control of an operator. So altogether, it's called an operon. So you can have several examples of this. For example, the lac operon, or the trip operon, or the arabinose operon. So these operons, they help in the transcription of related genes, because in prokaryotes, their mRNA is polycistronic. Poly meaning marami, as in a lot, and cistron meaning genes. So in prokaryotes, you have a single mRNA, but it can contain 3 genes, like this example here. So that even if you only have 1 transcription, you can have an expression of 3 genes. And so that would mean you will get 3 proteins from that single transcript. Okay. Now let's move on to transcription in eukaryotes. So in eukaryotes, you also have a promoter region. This thing here, the INR, is the transcription start site. And upstream of the transcription start site are these AT-rich regions, which are known as the Tata box. So the Tata box is evolutionarily conserved, such that you can see it in adenoviruses, in chicken albumin, in mouse, or in rabbit. So if you say that it's evolutionarily conserved, ibig sabihin, this region has existed for a very long time, and it has not been changed because it was seen to be advantageous to the organism. So in eukaryotic genes, we not only have the Tata box. Upstream of that, we also have the GC box, the CAAT box, and an octamer. Now, these parts here, these parts altogether are known as the extended promoter. But if we actually think of the core promoter, it will just be this side. So if you only have the core promoter, transcription will still occur, but if you have all these other sequences too, it helps enhance transcription. So if you see symbols like these, they actually mean transcription. So in eukaryotes, it is important for several regulatory mechanisms to govern transcription. So here, eukaryotes have 3 polymerases. We have RNA polymerase 1, 2, and 3. So RNA polymerase 2 is the one that is responsible for mRNA, whereas RNA polymerase 1 is responsible for rRNA, if I'm not mistaken, and 3 is for tRNA. So we're going to focus on RNA polymerase 2, which is for mRNA. In eukaryotes, transcription involves transcription factors. So transcription factors are proteins, and they help with transcription. The mnemonics that you have to remember here is that if you see the term TF, that means transcription factors. If you see this, transcription factor 2, that means these are transcription factors for RNA polymerase 2. And then, if you see a letter, transcription factor 2D, this means this is the sequence of when this protein was isolated. So, here, the mnemonic is DABFE. As in, if you have your DNA, and this is the Tata box, the Tata box is recognized by TF2D. TF2D has a subunit called TBP, as in the Tata box binding protein. So the Tata box binding protein recognizes the Tata box, obviously, and so it recruits TF2D there. TF2D next recruits TF2A and TF2B in no particular order. So it can be A first or B first, whichever, but the point is both of them are recruited on site. TF2A helps stabilize TF2D. TF2B also stabilizes TF2D binding, and B recruits F. So TF2F is attached to polymerase 2. As such, if you recruit F, you also help position RNA polymerase 2. Once those have bound, E and H come in. So TF2E and TF2H. Once they have attached to polymerase 2, you now have your pre-initiation complex. So, during this point, the DNA strands start to separate. So now you have an open complex. And the tail, so this is the tail of your polymerase 2, which is in the C-terminus domain. This tail gets phosphorylated, meaning phosphate groups are attached to it. So once it's phosphorylated, it is now activated. Once the polymerase 2 has been activated, B, E, and H dissociates. And these are eventually degraded. So, once they dissociate, your polymerase 2 zips through the DNA and produces the RNA. At the end of the sequences, at the end of transcription, polymerase 2 and mRNA dissociates. Your polymerase 2 here has to be dephosphorylated, meaning you remove the phosphate groups so that it can do another round of transcription. So, if you will find the transcription factors are important in positioning the polymerase 2 so that you will know where to start transcription. And they are also important in setting up the pre-initiation complex or in preparing for transcription initiation. Because if you lack one of these transcription factors, transcription will not occur. So this is one of the regulatory mechanisms in eukaryotic transcription. So I'm going to show you a video of eukaryotic transcription.
Transcription is the process of making RNA from a DNA template. Several key factors are involved in this process, including DNA, transcription factors, RNA polymerase, and ATP. Transcription begins with a strand of DNA. It is divided into several important regions. The largest of these is the transcription unit. This portion of the DNA will be used to produce RNA. Upstream of the transcription unit is the Tata box. An enhancer region may also be involved. Several complexes, known as transcription factors, are required for successful transcription. The first is TF2D, the largest of the general factors. A component of this factor, TBP, binds to the DNA using the Tata box to position TF2D near the transcription initiation site. Other transcription factors, including TF2A and TF2B, then attach.
So if you see, TF2A stabilizes the binding of TF2D, and TF2D contains the Tata box binding protein. And this is TF2B, as in baboy.
These complexes prepare the DNA for the successful binding of RNA polymerase.
So this is RNA polymerase, and this is TF2F.
Once RNA polymerase is bound, other transcription factors complete the mature transcription complex.
There. This is TF2E and TF2H.
Now, energy must be added to the system for transcription to begin.
So this is your ATP, and they are going to phosphorylate the polymerase 2.
This energy is provided by the reduction of ATP into ADP and PI.
So that's polymerase 2. Open
RNA
complex.
polymerase then synthesizes an RNA template from the strand of DNA. Most factors are released after transcription begins. When the end of the transcription unit is reached, the RNA polymerase dissociates and the newly formed strand of RNA is released.
And that's the end of our discussion for transcription. We're going to discuss post-transcriptional modifications in the next video.